Welcome to New Insights in Gene Fusions in GIST. Please remember the information provided in this web seminar is not intended as a substitute for your physician's guidance and care. My name is Sarah Rothschild, the LRG Senior Director of Program Operations, and I am your moderator for today. I would like to introduce our presenter today, Dr. Jason Sicklick. He's an Associate Professor of Surgery at the UC San Diego Moores Cancer Center. He is board-certified general surgeon and surgical oncologist, specializing in the treatment of sarcomas, including gastrointestinal stromal tumors, GIST. He received his medical degree from UCLA School of Medicine, and he completed his general surgery residency at the Johns Hopkins Hospital. He's on many committees, including the National Cancer Institute's GIST Working Group, and we also had the pleasure of having him at our LifeFest meeting last year in Torrance, California, where he was awarded the Clinician of the Year. We're very happy to have him present today on gene fusions, and I would like to have him take over and begin. Thank you, Jason, for joining us.
Good morning, good afternoon, everybody, and thank you very much, Sarah, for the introduction, and thanks for the opportunity to present some work that we've been doing, as well as others, on new insights into gene fusions in GIST. These are just my disclosures. I have funding from Foundation Medicine, which I will talk about a little bit, as well as from Novartis. So I want to start off by just discussing some basics of genomics in all tumor types. So some background, we have 46 chromosomes, so 23 sets of chromosomes, one set that we get from each one of our parents. And really these chromosomes and subsequently the underlying DNA within these chromosomes really is what is the backbone of the gene information that we all carry. Essentially we're all made up of combinations of A's that connect with T's and C's that connect with G's within our DNA. And merely sometimes just a single change in an A to a T or a C to a G or vice versa might potentially be the underlying reason driving cancers. So there's many reasons why we develop cancers. There's carcinogens, for instance, smoking, viruses and infections like human papillomavirus, which is associated with cervical cancer, radiation exposure, whether it be from medical therapies or from historical events like the Chernobyl nuclear meltdown have been associated with thyroid cancer as well as sarcomas, or mistakes in DNA copying, which sometimes can be hereditary and associated with mutations in some genes that are important in repairing or sort of correcting the typos, if you will, within our DNA repair. Or they can be spontaneous. But together, these are many of the factors that can lead to cancer formation. So these altered genes are often at the root of malignancies that people develop. And these genes code for many different types of proteins that can become altered. And then the resulting alterations can lead to activation in different signaling pathways. And for instance, you see here the RAS, RAF, MAP kinase pathway, which ultimately leads to cancer formation. So as I told you before, we have 46 chromosomes, but we carry about 20,000 genes in our entire genome. Some of the common names that you may be familiar with in the GIST community include names like KIT and platelet-derived growth factor alpha, BRAF, the RAS family, including KRAS, HRAS, and NRAS, as well as you're going to hear more about today about the FGFR1 or Fibroblast growth factor receptor 1. So some additional background on different types of alterations in cancers. So we normally, as I said, we have 46 chromosomes, and we have within the chromosomes we have genes that are built on this DNA platform. Sometimes we can get changes in the copy number, meaning that there's more copies of a given gene, as you can see here. We can get substitutions or what's called missense mutations, where a single base pair, being an A, a T, a C, or a G, gets changed to something else. We can have insertions and deletions, meaning that a portion of the gene is added in there or a portion is taken out. Or the thing we're going to focus mostly on today is rearrangements and fusions, where a portion of one gene might get swapped with another gene, whether that be on the same chromosome or on different chromosomes. Now, historically, the treatment for many cancers has been systemic chemotherapy, being drugs that basically target dividing cells, but often these are nonspecific. And as many of you know, GIST served as the paradigm for developing targeted therapies with the development of imatinib as a personalized treatment against the KIT oncogene. Now, historically, the testing for mutations within GIST, as well as many other cancers, has been based upon single or hotspot marker testing, where single gene mutations are evaluated on a individual basis. So many of you have probably had your tumors tested for genes including KIT and PDGFR alpha, and these were often done with specific focus just on those genes. But what's evolving over the last several years as technology has been rapidly changing is comprehensive genomic profiling platforms, which are now being clinically used, as well as being used on the research side. Now, some of the benefits to these being is that you can look at hundreds of genes all at the same time without any particular discrimination for a single mutation that you might be looking for, or a specific gene that you might be looking for beyond the known genes that you're initially would be potentially screening for in a hotspot. Additionally, there's the potential that you might be able to spare tissue because all of this studies for 400 genes, for instance, could be done on only a small fraction of tissue, as opposed to often the hotspot testing is based upon checking one gene and using some tissue for that, and then when that's negative, checking for another gene. And sometimes this can result in exhausting available tissue for mutation testing. So as I mentioned before, we're going to talk about some of the genes related to GIST. So about 85 to 90% of GIST are known to have driver mutations in several genes that most of you have probably heard of before. So the most common one being the KIT proto-oncogene, and the second most common being platelet-derived growth factor alpha. And what we know is that these can then lead to activation of downstream signaling pathways, including the RAS pathway. Now, the RAS pathway can lead to the activation of signaling downstream, including this MEK and ERK proteins. But even within this MAP kinase pathway, there can be mutations in some of these genes that can also lead to GIST, including RAS, as I mentioned before, BRAF, and NF1, the NF1 or neurofibromatosis 1 gene. Beyond these genes here on the left side, you can also see that mutations can occur in the succinate dehydrogenase subunits, SDH A, B, C, and D, as well as more recently we're now understanding that you can develop methylation changes within the promoter region, or the region that's controlling the transcription or the production of RNA from a gene, that can then lead to alterations in the genes themselves. But there still remains a subset of GIST that we don't know a whole lot about in terms of what the mutation is, and these have been termed quadruple wild-type GIST. There was a study just published just less than 2 years ago looking at these quadruple wild-type GISTs, and basically at that time we knew very little about them. We knew that many of them had expression of the succinate dehydrogenase B protein, but they lacked the insulin growth factor 1 receptor. But there was very little knowledge known about the age, sex, site, or multifocality of these tumors. Moreover, we didn't know much in terms of the genomics or the epidemiology or the disease biology of these tumors. So one of the things that our lab started looking at a couple of years ago was to try to implement broad genomic profiling, or this comprehensive genomic profiling, to evaluate quadruple wild-type GIST to see if we could understand more about the genomic alterations and the disease biology of this patient population. And I'm not going to belabor the details here, but this was work was done in collaboration with Foundation Medicine, and we looked at patients from 2012 to 2015, and we used their technology to evaluate at the minimum 315 cancer-related genes, as well as 28 genes that have been associated with gene fusions. And we did subsequent data analysis to look at not only what were the known mutations within these tumors, but also what have been termed variants of unknown significance. And for some of you that may have seen reports from some of the different companies that do genomic profiling, the results sometimes include these VUSs, or variants of unknown significance, that may or may not be associated with cancer. But one of the things that we did in our analyses was to look at the mutation, or these alterations or VUSs, to assess whether they may be deleterious or may be cancer-causing based upon several different prediction modeling programs available. And you can see there in the middle of the screen the SIFT program, this polyphen, the mutation taster, and as well as mutation assessor. But to suffice it to say, what we show, the data I'm going to show you in terms of some of the genes that we've evaluated, we elected to only include genes that had at least 2 of these different programs suggesting that these may be deleterious or cancer-causing. And so from here on in, I'm going to focus on genes that are known to cause cancers, as well as genes that are highly likely to be associated with cancer formation. So the first thing I'll show you is a plot of patients as well as the genes. So across each one of these columns is a different patient. And downward are all the genes associated with their tumors. And the first thing that you'll see is there on the top line is that there are KIT mutations. And I don't want to really focus on each individual name of these genes, but the point to take away from this graph is that GIST patients, and this is all GIST patients in our study of 186 patients, have a very heterogeneous makeup of the genomic landscape of their tumors. And no 2 patients are exactly alike in terms of mutations that are associated with their tumor formation. If we look at the common mutations that were found in these patients, as not to be unexpected, about 70% had KIT mutations, 12% had platelet-derived growth factor alpha mutations, 10% had NF1 mutations, 8% had SDH subunit mutations, 4% had BRAF, and then the remaining 2% had KRAS. But there remained 12% of patients that did not have mutations in any one of these known genes. Now, half of these patients that we looked at were analyzed prior to SDH being included within the gene panel that Foundation Medicine was using, and then the second half was included SDH mutation testing. So the first things that we will note if we look at this patient population is that there's about 162 patients that were non-wild type, if you will, and 24 patients with wild type. Now the average age of these patients was in their mid-40s for the wild type patients. You'll see there the mean age is 44 for those that were negative for all genes tested, and then there were the average age was 44 for those where they were negative for the known driver mutations in GIST, but prior to the testing for succinate dehydrogenase. The next thing I'll point out is that many of these tumors tended to be larger tumors, as you can see here in the quadruple wild-type group. And a vast majority of the triple wild-type tended to have no lymph node metastases, and about a quarter of patients in the quadruple wild-type group had lymph node metastases. Now, if we go back to the genetic work that we mentioned earlier in terms of the different types of mutations, I've put a little, just a reminder of the different, a picture of the different types of mutations that we were seeing. Now, not unexpectedly, the majority, about 70% of the mutations that we saw were missense or substitution mutations, as you see right here. But we also saw indels or insertions or deletions. And if we move all the way down to the end, we saw several cases of gene fusions. And these are these rearrangements and gene fusions over on the far right that we're going to really focus on for the majority of the rest of our talk. Now, when we looked at the actual breakdown of these patients, this is a similar plot to the one you saw earlier, with each patient being a column and each tumor, being a cross, and each gene going down. And when you can start with more granularity here, you can start seeing that there's clearly also a heterogeneity within this population of patients. The ones on the left being those that were the quadruple wild type, and the ones on the right being those that were negative for all genes with the exception of not being tested for the succinate dehydrogenase subunits. Now, if we look further, what we notice is that in our analyses, it was that there were 7 genes that were more commonly affected in the patients that were wild-type patients as opposed to the non-wild-type patients. And you can see here on the highlighted right side, these alterations were fairly common in this subset of patients. And these were all statistically significantly more frequent in this patient population. Now, one of the genes that popped out of interest was this FGFR1 or fibroblast growth factor receptor gene. And what we found in our study was that the FGFR1 gene had gene fusions in 2/3 of the GIST patients with FGFR1 alterations. And so for the first time, what we found is that this FGFR1 gene had actually been spliced to 2 other different genes. One being this HOOK3 gene, which is a gene that's associated with vesicles within cells that traffic proteins. And then the second gene that it was associated with is this TAC1 gene, which is partially associated with cell division. So here we are showing that these hybrid genes basically are forming from two separate genes. And just to provide you a little bit more insight into how these different types of fusions can form beyond what I showed you earlier in that animation, is sometimes these mutations or these fusions can form from different chromosomes. So you can see here on the left you have chromosome number 1, and then on the right chromosome number 2, where there's basically a flip-flopping of the genes so that you've got this fusion between these two, creating the orange to the green. There can also be situations where the middle portion of a gene gets completely cut out, and so gene 1 and gene 2 become fused together, and this is the case what can occur in the FGFR TAC1. And then there can also be flipping, essentially, or chromosomal inversion, where one part of the chromosome actually flips up. So this darker green can flip up and then join up here, as opposed to the light green being closer to the orange gene. So we presented this work about 1.5 years ago now in Salt Lake City. And at the same time, besides these FGFR1 fusions, the other fusion that we identified was an ETV6 NTRK3 fusion. And why these are all important is because all these gene fusions have been reported to be cancer-causing in many other tumor types. And you'll see here in the table on the bottom of this slide that the FGFR1 TAC1 fusions have been reported in glioblastoma multiforme, which is a very aggressive form of brain cancer. It's also been reported in breast cancers as well. The FGFR1 hook 3 has been reported in patients with papillary thyroid cancer. And then down below on the bottom, the ETV6 NTRK fusion has been reported in infantile fibrosarcoma as well as breast cancers and salivary gland tumors. So at the same time that we had presented our data on the ETV6, the data was also reported by a group in Italy, Branca's group, looking at a patient that they had identified that was a quadruple wild-type GIST patient. And in this study, they also, similar to us, found that there was an ETV6 NTRK fusion, very similar to the one I showed you in the earlier slide. And again, they essentially saw the same portion of the ETV6 gene fusing with the same portion of the NTRK gene. Now, at the same time, Michael Heinrich and Chris Corliss at OHSU evaluated their patient population with 5 patients that were quadruple wild type, some of these being provided through samples through the LifeRAF group. And in their analyses, when they looked at the gene fusions that I briefly showed you here in this table, what they found is they also identified one patient with an ETV6 NTRK3 fusion, as well as a patient with an FGFR1 TAC1 fusion. In their panel, they did not include the hook 3, so it's unknown whether potentially these later patients potentially could have had FGFR1 hook 3 mutations. And again, these were fairly similar gene fusions to the ones that we'd identified in the original cohort of patients we'd identified, as well as the one that Branca's group had identified in their analysis. Now, looking at the data from Branca's publication in the Journal of Pathology, they went on to develop or clone, if you will, the gene fusion that they had identified in their patient. They then took this portion of the gene and actually put it into 2 different cell lines, both an infantile fibrosarcoma cell line and an osteosarcoma or bone sarcoma cell line. And what they found in their study is that in those tumor cell lines that express the mutation that they had seen in their patient with GIST, that it sensitized the tumor cells to both IGF-1R inhibitors as well as ALK inhibitors. So suggesting that there's potential for therapeutic targets within these gene fusions. Now, to provide you a little bit more background on the NTRK gene, this is also known as the Neurotrophic tropomyosin receptor kinase. Now, what becomes important, as you'll notice here, is that this is a fairly similar in terms of its biology to KIT, PDGFR alpha receptor tyrosine kinases. You'll see here that, similar to what I pointed out earlier, that the NTRK genes, or the NTRK proteins I should say, can lead to activation of the RAS, RAF, MEK, ERK, or MAP kinase signaling pathway. They can also lead to other signaling pathways, including the PLC protein pathway, as well as the PI3 kinase AKT pathway. So over the last couple of years, there's been several drugs that have been developed that are being studied as potential NTRK inhibitors. And I'm going to focus on one of those being the drug developed by the Loxo company called Loxo 101. I'm picking this specifically, this gene expression, this drug, based upon subsequent work that we've done in collaboration with the group at MD Anderson. So at the same time that we were presenting our data at the CTOS meeting, Dr. David Hong from MD Anderson was presenting data at AACR, American Association for Cancer Research meeting, with treatment of the patient with the ETV6 NTRK gene fusion that I mentioned earlier in our patients, in our 186 patient data set. And what this patient had been treated with 5 different lines of therapy for their GIST and had progressed on all 5 lines of therapy. The patient was then found to have this ETV6 NTRK GIST gene fusion and was started on the LOXO 101 clinical trial being run at MD Anderson. And what you'll see here on this PET scan is on the left side there's the basal PET scan where you can see very high uptake of the tumors. And on the bottom here you can see a large tumor within the liver with uptake. And this is after 8 weeks of therapy. You can see that there's a dramatic decrease in the amount of uptake of the tracer within the tumors throughout the patient's abdomen, as well as you can see here on the bottom that there's a significant reduction in the uptake within this large liver tumor as seen on the scan to the left here. Moreover, you can also notice that there's a significant reduction in the size of the tumor as well. So with that information, we're obviously beginning to hone in on potential therapeutic targets within these GIST gene fusion patients, and more specifically being the NTRK gene fusions. Now, if we look at going back to the fibroblast growth factor receptor gene mutations that we identified earlier, again, you'll notice some very similarities to both the NTRK as well as KIT and PDGFR alpha. And again, you can see that there's the RAS, RAF, MEK, ERK pathway, PI3 kinase, and the PLC. So again, you can see here that here was the NTRK picture I showed you earlier. Here's a example of some of the signaling pathways within activation by KIT and PDGFR alpha. So again, a lot of similarities between these receptors that are then activating very similar pathways associated with GIST formation. So to date, there are also FGFR1 inhibitors both in clinical trials as well as in clinical care at this point. And I just show you here a figure taken from paper published by Cato and colleagues in the International Journal of Molecular Medicine, just identifying several different compounds and drugs that are clinically available for targeting the fibroblast growth factor receptors. So what I've shown you over the last short bit of time is that we're really beginning to hone in on the information in these quadruple wild-type GIST patients. We're beginning to understand more about the genomics, the epidemiology, and the disease biology. And what is turning out is a lot of them have fusions or alterations in genes. So we've now identified that FGFR1 fusions can occur in these patients. We've identified ETV6 NTRK3 fusions in these patients, as well as a handful of other genes that I've got listed there that did not have fusions but have mutations of other types. We're also beginning to understand that these patient population tends to range in the 30s to 50s, tends to be a little bit more common in men than women, and that we're seeing these tumors in a fairly similar distribution to the distribution that we would classically think of for the GIST population in terms of occurring anywhere from the stomach all the way to the small bowel and colon. Now, one of the differences from the general understanding of most GIST tumors and GIST biology is that we saw fairly frequent lymph node metastases in a subset of these patients. Now, turns out there's actually more fusions now being identified in GIST. And this is a publication by the NIH Wild-Type GIST Clinic that was published by Sosi Patros Boykos and colleagues in JAMA Oncology last year. And they identified one patient with a KIT PDGFR alpha GIST gene fusion here that you're noting on this figure. So taking all these together, we can actually start looking at the human kinome. And the human kinome is basically a phylogenetic tree, if you will, cataloging all the known kinases within the human cell. And you can see here that there's different families based upon the colors here, as you see. But what it turns out is if we focus on up here, that many of these genes that we're talking about today, both starting with KIT and PDGFR alpha, which are fairly similar in terms of their phylogenetic tree, are also related to genes that we're talking about in terms of the new fusions that we're identifying in GIST. So beyond BRAF and the TK genes that were included in our genes associated with GIST, we're identifying that these fusions are occurring in the TRK as well as the FGFR genes, which are, as you can see here, are fairly similar in location on the phylogenetic tree of the kinome to KIT and PDGFR alpha. And also the NIH group has identified this KIT PDGFR alpha gene fusion. So what we're starting to understand is that within this portion of the phylogenetic kinome that we're identifying fusions within several of these genes which all have similar downstream mechanisms of activation. So to begin summarizing what we're finding now is we're now beginning to understand that GIST represents an increasingly heterogeneous disease. It's not just KIT mutations or PDGFR alpha mutations and then everything else. Every patient's tumor tends to be a little bit different in terms of its genetic makeup. And what we're now beginning to understand here is that there's not just two genes being KIT and PDGFR alpha, but perhaps there's at least 22 genes that we're identifying associated with GIST. These include the things like KIT and PDGFR alpha, as well as several other genes that we've identified in our publication that was recently reported in Journal of Translational Medicine, as well as genes reported by the NIH group, and most recently two genes, MAX and MEN1, that were reported a couple of weeks ago by Pantaleo's group from Italy. So the take-home message from this slide is not to start looking at all these different boxes here, but to understand that there's a heterogeneity to these tumors that we're just beginning to touch upon. If we look to summarize the GIST gene fusions that we've identified in both our patient population as well as within the literature, it looks like to date there have been 7 gene fusions reported in GIST. As you see here, the average age of the patients in all of these studies is about 50 years old with a median of about 54. So these are likely a different disease and a different disease biology than the wild type patients that are typically associated with the SDH patients or with the pediatric GIST population. We tend to see that these are more common in men than women, and that these can occur anywhere within the GI tract, namely being all the way located from the stomach all the way down to the rectum. The other identifications that we've noted are that about a third of these tumors have been associated with lymph node metastases, as well as about over 40% have been associated with distant metastatic disease. And then finally, to summarize the frequency of these mutations reported to date, so about 43% have been the ETV NTRK, similar amount in terms of the FGFR1, and then the one patient, or 14% of the 7 patients with a KIT PDGFR alpha mutation. So in summary, we're starting to better understand that these quadruple wild-type GISTs are really not wild-type. That it turns out that they do have mutations in other genes, whether these be these substitution mutations or the gene fusions that we've focused upon today, suggesting that really there still remains an unclassified set of GISTs that we don't understand the biology of, but that we're beginning to break down the barriers in terms of understanding the biology of these quadruple wild-type GIST patient population. And what we're beginning to learn is that these tumors frequently spread to lymph nodes, and that most of these are not truly wild-type. We're also identifying that there's potentially deleterious or cancer-causing gene fusions that are occurring in adult GIST patients, and that these are potentially treatable with drugs that are either in clinical trials or that are FDA approved. These include the KIT inhibitors, the NTRK inhibitors, as well as the FGFR1 inhibitors. So in conclusion, these findings really serve as a lead point for new research into GIST pathogenesis and truly open up a new potential for studying GIST gene fusions, as well as understanding how the biology of these tumors can be targeted with therapies to provide durable responses for patients. And with that, I would like to say thank you for the opportunity to discuss this today, and would be happy to answer questions. I'm going to leave this slide up here for a minute. If anybody wants to take down my contact information, I'd be happy to take emails at a later time.
Great. Thank you, Dr. Sicklick. Thank you for this presentation today. I know this is a new area for many people, and probably for most, it takes a while to sort of wrap our heads around this new material and the science behind it. So I really encourage folks to type in questions, even if you think it's basic, but just to really help you understand more about this concept, because it's a new emerging area, and I think it's important for you to understand, especially in terms of going back to your doctor if you think it's necessary to ask questions about it. So just to start off the questions, so it looks like from your presentation that the gene fusions might not be isolated to just the wild-type population, but could also be occurring in the KIT mutant GIST patients as well. Right, Dr. Sicklick?
Correct. To date, there's been one reported case from the NIH group of the patient with a KIT fusion to the PDGFR alpha. Beyond that, I don't obviously know how frequent this is. I would suspect it's probably maybe a little bit more frequent than just one case, but the true incidence is unknown at this time.
So how does one look into getting tested for gene fusions?
So in terms of testing, the first point to make would be is that most of the hotspot testing, as it's currently done, will not pick up the gene fusions unless somebody is specifically performing a test where gene fusions are the key question being asked. So there can be individual tests associated where, for instance, if you're specifically only looking for the ETV6 NTRK fusion, that could be evaluated for. But in the general sense of sending your tumor off to get its KIT mutation testing or PDGF or single hotspot testing, it's going to be missed there. And so in those scenarios, the ways to identify these fusions would be sending them off for larger testing panels at this time, because it's a little bit of a shotgun approach in terms of you don't necessarily know that the patient does or does not have a fusion. but you perhaps either know that they don't have a KIT or a PDGFR alpha mutation at that point based upon hotspot testing, or has become my practice over the last several years is that all patients that I see get comprehensive genomic panel testing with these large panels of genes that upwards of close to 400 genes in these panels, which include testing for a subset of gene fusions.
Okay, thank you very much. So just help us understand, because there was a question, is a gene fusion a special type of mutation?
It is in a sense. It's basically you're connecting two different genes together, where one gene is basically talking to the second gene and potentially helping tell it what to do, if you will. So it's a type of mutation, but it's different in the sense of what we classically think of in terms of a mutation being one of those A's, T's, C's, or G's gets changed to a different A, T, C, or G. That's not as it's supposed to be. But this is basically connecting two different genes together where they're talking between one another. And often what's happening is for instance, the FGFR gene is on in these tumors, and then it's creating a protein that's a mixed protein of both the FGFR1 and the TAC1, which is then leading to alterations in the biology within these tumor cells.
Okay, thank you very much. So if one finds out that they have a gene fusion, and you're saying that there are other targetable drugs out there, either in clinical trials or FDA approved, how does one decide what's the most appropriate choice for targeting that fusion?
So I think in those scenarios, the best situation would be is for them to talk to their treating physicians to figure out what potential therapeutic options are available, whether that be a clinical trial, which would be the ideal situation in rare disease like this, and in identifying new genes that are causing this, or potentially using off-label use of drugs is another option. But the first option is always a clinical trial if possible.
Can you explain the concept of biomarkers?
So a biomarker in a general sense is something that can be used as a surrogate for understanding the biology of a tumor. So, so these can either be markers on the tumor cell themselves, or sometimes circulating within the blood. But they're a snapshot, if you will, or information provided by the tumor that we then can look into to understand the biology of those tumors. As an example, in a lot of other tumor types, we talk about using tumor markers within the blood as a marker of the progress of a disease. So for instance, colon cancer, we use a marker called CEA, which is in the blood. And when that's elevated, we can follow that in terms of responses to therapy or recurrence of disease. And so it's variable, and it's really specific to each type of cancer. In terms of just the available biomarker that we have, which we use in terms of making the diagnosis and potentially treating patients is most commonly KIT, or the DOG1 gene, discovered on just one gene, which is used in the process of making the diagnosis of GIST. But we do not have any kind of blood biomarkers to follow this disease at this point.
Okay, thank you very much. How would a quadruple wild-type patient proceed with trying to send their samples for the larger testing panel to try to find a fusion? Is there a particular lab that could be contacted to do this?
So there's several commercial companies that do large panels of genes. The one that we've collaborated with, being my disclosure, is Foundation Medicine, and the data that I've shown you today from our group has been done in collaboration with Foundation Medicine. But there are other groups that have large gene panels that are commercially available as well. In addition, depending upon which institution you're being treated in, there are many institutions around the country now that have their own gene panels. Here at UC San Diego, we have a gene panel that has been developed that has about 400 genes in it, which include gene fusions as well. So it's sometimes not too far from home that these testing may be done. And this is often available just using the tissue blocks that are the formalin-fixed paraffin blocks that should be available from either a biopsy or from the time when a tumor was removed, that can then be used for extracting the DNA to then do the testing.
Thank you very much. Of these gene fusions, are they inheritable? Are c-KIT or PDGFRA inheritable? Or are only the SCHNF1 inheritable?
So to my knowledge, the gene fusions are not inheritable. In terms of the mechanism why they occur, there's probably, most of the time, it's some random event where the editing, if you will, but there's essentially one paragraph in your page gets removed, and so two paragraphs that don't belong together get put together. That's essentially what's happening with these fusions. Now, in terms of, as you mentioned, the SDH patient population does have a heritable form that can be passed from parent to child. There are a few families that have been reported to have germline, meaning that the parents have it and give it to their children, mutations in both KIT and PDGFR alpha, but it's exceedingly rare. To my knowledge, I believe there's only 3 families that have been reported with KIT, with germline KIT mutations that have transmitted to family members, or I believe only one PDGFR alpha family reported. So there's potentially more out there that haven't been reported in the literature, but it's exceedingly rare relative to the frequency of just spontaneous events occurring in patients.
Thank you. I know that we have some representatives from other countries around the world, and I wanted to know what could they do to create more awareness about this concept of gene fusions in GIST?
That's a great question. I think that trying to provide education to both other patients as well as doctors through groups like LifeRaft is a critical way to spread the knowledge for patients both in the United States as well as in other countries. This is obviously an evolving field, and it's also a small fraction of the larger group of GIST patients. So it's a matter of education through groups like LifeRaft, through reading the literature that outside other physicians as well as other individuals in other countries will become increasingly aware of this. And to speak of this type of webinar being set up by LifeRaft is clearly another great way to spread knowledge about new findings such as the gene fusions in GIST. Thank
you, Dr. Sicklick. We really appreciate the time today to present to the GIST community and answering these questions. I'm sure that it's going to take a bit for people to sort of absorb the science behind it, and they'll probably check back into the recording just to re-listen and relearn. But we thank you for making yourself available to be contacted more about this subject, and we hope that we can continue to provide more information and education about this area as it is new and emerging. So I want to thank everyone for joining today, and I also want to thank Dr. Sicklick for presenting. It was excellent. Thank you very much.
Thank you very much.